Hello everyone, this is group number 5. Our topic is COVID-19 data analysis and I am the group representative Chetna Rao. And the rest of our group members are Himashvi, Priyanka, Soma, Shrujana and Shriusha. In this project, we will show how to do data cleaning and pre-processing, data visualization, data statistics and hypothesis testing. In this project, we are going to work with the COVID-19 dataset published on GitHub. After Also, we have another data set consisting of deaths due to oxygen shortage and non-virus deaths. We are going to merge these two data sets to see if there is any relationship between the spread of the virus. The coronavirus or COVID-19 is a large family of viruses that causes illness ranging from the common cold to acute respiratory syndromes. Common symptoms of the novel coronavirus strain include respiratory symptoms such as fever, cough, and shortness of breath, according to the WHO. The WHO has declared the coronavirus epidemic as a global health emergency. The libraries used in this project are NumPy and Panda for data interpretation and Matplotlib, Seaborn, Plotly for data visualization. We will do the project by following these steps. First, we have to import COVID-19 dataset and prepare it for the analysis by dropping columns and aggregating rows. Second, deciding on and calculating a good measure for our analysis, merging two datasets and finding correlation among our data. Fourth, visualizing our analysis results using Seaborn.
Data preprocessing and cleaning. The main objective of the data cleaning and preprocessing is to understand the techniques such as lambdas and manipulating CSV files, and to describe common Python functionality and features used for data science, and also to clean and process data frame structures by querying them. Libraries used in data cleaning process are Pandas and NumPy. Pandas, it has a built-in data structure that makes manipulation and analyzing data collection easier. NumPy introduces multi-dimensional arrays and matrices as well as functions that make complex mathematical and statistical operations on these objects simple. About data frame. A data frame is a two-dimensional labeled data structure with multiple sorts of columns. These are some of the methods of data frames here shown in the slide. Methods used for cleaning data: splitting, imputation, and interpolation. Splitting. Pandas sample is used to produce a random sample row or column from the data frame function. Imputation. Imputation is the process of replacing missing data with substituting values. Interpolation. Interpolation is a method of building new data points within the range of a discrete collection of known data points, and it is a sort of estimation. Preparation data for consumption. While preparation of data for consumption, first of all we have to clean data and group the related data. After that, prepare the data in order to perform a good performance. These are the libraries we have used for the data cleaning and analysis of COVID data. These are some data files we used in COVID data analysis. The files are read by using Panda libraries as shown in the slide. Cleaning data of COVID cases in India. This is a raw data of given file COVID cases in India. During cleaning process, we have to remove unwanted columns. To reset the index, here we are removing the column serial number by using drop function. After that, to get the total number of cases, we are adding total confirmed cases in India and Indians who are living in foreign nation. Now, to get the active cases, we are subtracting deaths and cured cases from the total cases. In this way, we are preparing data. Now, we have to check whether null values are there or not in the given file. To check the null values in the data, we have to use isNull function as shown in the slide. If in case null values are present in the given data, we have to replace them by using the data frame.
Now we are going to do data visualization for the data set of COVID cases in India. The main objectives are these, and these were the libraries used to do visualization. This graph depicts the percentage of total cases and active cases in India as a pie chart. To create this pie chart, we need to import a library called Matplotlib PyPlot. Thus, we first imported it as PLT and then calculated the average of total cases and active cases in India. With this information, we create a pie chart that look like this, with the red color indicating the total number of cases and the yellow color indicating the active number of cases. We may modify the color to our desired by changing the color name here. Let's have a look at the total data's graphical representation now. This is the bar graph of total COVID cases in various Indian states. As can be seen, Maharashtra has the highest number of oral COVID cases when compared to other states, owing to a variety of variables such as its large population. This is the bar graph depicting the total number of deaths in different states. As the number of cases was higher in Maharashtra, the number of deaths was also higher in Maharashtra. This graph depicts the total number of active cases in different states. The scatter plot depicts the total number of cases in different states. Now we will look at how the coronavirus is spreading over the world. For this, we looked at trends in total cases and new cases in India and around the world for a specific time period, for example 100 days, and made conclusions from that. This is the data for India's per day cases. In this data, we won't need the days after suppressing 100 cases column in our analysis. Therefore, we will remove it with this function. Using this function, we rename total cases to total cases in India and new cases to new cases in India to avoid confusion. And now we are going to compare the trends of COVID cases in India with other countries. For that, we took some reference countries like Italy, Korea, and Wuhan. Similarly, we did a cleaning and processing for data set of Korea, Wuhan, and Italy. This is the final data set of Italy, Korea, and Wuhan after cleaning and processing.
Hello everyone, myself Amanikmar. Now we are presenting data visualization of the data file COVID case in India. Now see this graph. This is a graph which shows the change in total number of cases of Indians day by day for certain period of time using plotly method. Now see this graph. This is a bar graph of same data but plotted with another method. Now we will talk about data visualization of the data file per day cases in India. This graph shows day-wise total number of confirmed COVID cases in India. Now we will discuss about the data visualization of COVID cases from other countries. Now see these following graphs. This graph shows the day-wise total number of confirmed cases in Italy. Now see following graphs. This graph shows the day-wise total number of confirmed COVID cases in Korea. This graph shows the day-wise total number of confirmed COVID cases in Wuhan. And this is the Python code to plot the graphs of day-wise changes in confirmed cases in 4 countries. And these are the graphs of 4 countries. Thank you.
Worldwide analysis of the data. Here we are reading the COVID-19 data file, and this is the data of the file. In this data, we have dropped serial number column and renamed some of the column. After that, we have grouped the date column as shown in the slide, and also grouped date with respect to confirmed cases, deaths cases, and recovered cases individually for the sake of data visualization as shown in the slides.
Now we will visualize the data for different countries between Jan 2020 to March 2020. This is the plot of worldwide confirmed cases versus date. Here, as the date increasing, confirmed cases is also increasing. This is the plot of worldwide deaths versus date curve. This is the plot of worldwide recovered cases versus date. Here, we are plotting all above three curves together. This is the code for below curve. This is the graph of countries recovered death and confirmed cases. Here we got the information that China is the country where maximum number of recovered and confirmed cases were found between Jan 2020 to March 2020. Now data visualization for oxygen shortage death and non-virus death in India. This is the data set of a state-wise oxygen shortage death in India. This is the data set of a state-wise non-virus death in India. This is the plot of oxygen shortage death versus a state in India. Here we can clearly see that Karnataka is the state where maximum number of oxygen shortage deaths were found. Now we are trying to plot a pie chart of oxygen shortage death and non-virus death. Here we got oxygen shortage death 25% and non-virus death 25%. So we can say that in India, number of cases death due to non-virus death is 3 times larger than the number of oxygen shortage death.
Data statistics. In this part, we have sorted the states in ascending order based on the number of deaths
and we have observed that Maharashtra has recorded maximum number of deaths. And again we have sorted the states in ascending order based on the number of active cases, and we have observed that Maharashtra has recorded maximum number of active cases. And when we have sorted the same states in ascending order based on the number of total cases, we have observed the same Maharashtra has recorded maximum number of total cases. Data hypothesis. In this part, we have used a statistical theory named null hypothesis. The main objective is to find the nature of the data plot. For that, we want to find whether the plot is normally distributed or not, or whether the plot is right or left skewed. The libraries we have used are Sci-Fi library. In India, for plot of total cases versus date, we assumed that the plot is normally distributed. Here when we write code, we got p-value less than 0.05. That is, null hypothesis is rejected. That means the plot is not normally distributed. For the same plot of India, we assume that the plot is right skew. Here when we write code, we got mean of the data is more than the median. That means the null hypothesis is accepted. That means the plot is right skew. When we do the same thing for Italy, we got p-value less than 0.05. That means the plot is not normally distributed, and at the same time it is right skew. When we do the same thing for Korea, we got p-value less than 0.05. That means the plot is not normally distributed, and at the same time it is right skew. When we do the same thing for Wuhan, we got p-value less than 0.05. That means the plot is not normally distributed, and at the same time it is left skew. Thank you. Thank you for your attention. That's the end of our project.